outbidding devices topics I left yet to cover. So let's continue and finish it today by itself. So let's start without further delay. Outbreeding devices. Majority of the flowering plants produce hermaphrodites. ज़्यादातर plants hermaphrodites हैं. ज़्यादातर plants hermaphrodites flowers produce करते हैं and pollen grains are likely to come in contact with the stigma of the same flower. Continued self pollination resulting in inbreeding depression cause कर देती है. Flowering plants has developed many devices to discourage self pollination and to encourage the cross pollination. In some species, pollen release and the stigma become receptive are not synchronized. Either the pollen is released before the stigma become receptive or the stigma become receptive much before the release of the pollen tube and pollen tube. In some other species, the anther and the stigma are placed at the different positions hypergamy से बोलते हैं so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent करते हैं orthogamy को. Receptivity वाला and position वाला ये दोनों orthogamy को prevent करेंगे. The third device to prevent the inbreeding is the self-incompatibility. That is, this is a genetic mechanism and prevents self-pollination from the same flower or the other flowers of the same plant from fertilizing the ovule by inhibiting pollen germination or the pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unicellular flower. If both male and female flowers are present on the same plant, such as the castor and maize, that is, they are monoecious, that male and female flowers are present on the same plant. मेल फ्लावर फीमेल फ्लावर सेम प्लांट में प्रेजेंट है. सच कंडीशन देखने को मिलती है कैस्टर और मेज में और ये मोनोशियस प्लांट है. अगर मेल और फीमेल एक ही बॉडी में है फ्लावर्स तब ये कहलाएंगे मोनोशियस प्लांट. It prevents autogamy but not the geitonogamy. In several species such as the papaya, male and female flowers are present on a different plant. That is, each plant is either male या तो female. ऐसी कंडीशन को हम डायसी कंडीशन बोलते हैं. This condition prevent करती है both autogamy और geitonogamy को. Pollen pistil interaction की बात करते हैं. Pollination does not guarantee the transfer of the right type of pollen. Compatibility pollen of the same species as the stigma, often pollen of the wrong type either from the other species or from the getting sleepy. But have to read this completely. Just focus. Often pollen of the wrong type either from the other species or from the same plant. If it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of the right type compatible or the wrong type incompatible. If it is the right type, the pistil accept the pollen and promote करती है post pollination events को. Post pollination events that leads to fertilization. If the pollen of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or the rejection is that result of the continuous dialogue between the pollen grain and the pistil. This dialogue is mediated by the chemical components of the pollen interacting with those of the pistil. It is only in the recent year that the botanist has been able to identify some of the pollen and pistil components and the interaction leading to the recognition followed by acceptance or the rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the pollen grain germinates on the. It is only in the recent period botanists has been able to identify some of the pollen and pistil compatibility and interaction leading to the recognition followed by the acceptance or rejection. As mentioned earlier, flowering compatibility pollination, the pollen grain germination on the stigma to the produce a pollen tube through one of the germ pores. The content of the pollen grain move into the pollen tube grow through the tissue of the stigma and style and reach the ovary. You would recall that in some plants pollen grain are shed at the two condition, that is the vegetative cell and the generative cell. In such plants the vegetative cell divide and form the two male gametes during the growth of the pollen tube in the stigma. In the plants which shed pollen in the three-celled condition, pollen tube carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enter one of the synergids through the filiform apparatus. Many recent studies have shows the that filiform apparatus present in the micropylar part of the synergid guide करती है entry of the pollen tube को. All these events from pollen deposits on the stigma until the pollen tube enters the ovule are together termed as the pollen pistil interaction. Pollen pistil interaction, as mentioned out earlier, pollen pistil interaction is a dynamic process involving the pollen recognition followed by the promotion or the inhibition of the pollen. The knowledge gained in this area would help in the plant breeders in manipulating pollen pistil interaction, even in the incompatible pollination to get desired hybrid. You can easily study pollen germination by distinguishing some pollen from the flowers such as pea, chickpea, Crotalaria, basil, and vinca on a glass slide containing a drop of sugar solution about 10% after about 15 to 30 minutes. Absorb the slide under the low power lens for the microscope. You are likely to see pollen tube coming out of the pollen grain. As you shall learn in the chapter on plant breeding, a breeder is interesting in crossing different species and often generate to combine desirable characters to produce commercially superior varieties. Breeder is interested in crossing different species and often generate to the combined desired character to produce a commercially superior variety. Artificial hybridization is one of the major approaches to the crop improvement program. In such programs experiment, it is important to know make sure that only the desired pollen grain are used for the pollination and the stigma is protected from the contamination from the unwanted pollen. It is achieved by the emasculation and the bagging technique. If the female partner be a bisexual part, नहीं that was good. If the flower female parent pair bisexual flower, removal of the anther from the flower bud before the anther discs using a pair of the forceps is necessary. This step is referred to as the emasculation. Emasculation flower has to be covered with a big bag of the suitable size, generally made up of the butter paper, to prevent the contamination of its stigma and the unwanted pollen. This process is called as bagging. Right, Jono? Bagging. When the stigma of the bagged flower attain hmm, सही है. Bagging. When the stigma of the bagged flower attain receptivity, mature pollen grain collected from the anther of the male partner are rested on the stigma and the flower are rebagged and the fruits allowed to develop. If the female parent produce unisexual flower, there is no need to be emasculation. The female flower bud are bagged before the flower open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower is rebagged. Now it's the topic of double fertilization. With that, we have a seed topic and then apomixis and polyembryony. Three topics are left over that. 2 3 pages. So we have to be completed within the half an hour. So let's continue from the double fertilization. After entering one of the synergies, the pollen tube releases the two male gametes into the cytoplasm of the synergids. Cytoplasm of the synergids. One of the male gamete move towards the egg cell and fuse with the nucleus and complete the syngamy. This result in the formation of a diploid cell. The zygote other male gamete move towards the two polar nuclei located in the central cell and fuse with them to produce a triploid primary endosperm nucleus. As this involves the fusion of three haploid nucleus, it is termed as a triple fusion. Since two type of fusions in gamete and the triple fusion take place in an embryo sac, the phenomena is termed as the double fertilization, an event unique of the flowering plants. The central cell after triple fusion become the primary endosperm cell and the ovary and develop into the endosperm with the zygote develop into the embryo. Hotspot. ओके. यहाँ तक हो गया. अब बात करते हैं पोस्ट फर्टिलाइजेशन स्ट्रक्चर्स एंड साइटोप्लाज्म ऑफ सिंगेमी में डिपॉजिट होते हैं. पोस्ट फर्टिलाइजेशन स्ट्रक्चर्स एंड इवेंट्स की बात करते हैं. तो हमारे पास यहाँ पे following double fertilization events of the endosperm and the embryo development, maturation of ovule is into seed and ovary into the fruit are collectively termed as the post fertilization events. Endosperm. Endosperm develop precede endo precedes embryo डेवलपमेंट एंडोस्पर्म डेवलपमेंट करने के बाद एम्ब्रियो का डेवलपमेंट स्टार्ट होता है वाई द प्राइमरी एंडोस्पर्म सेल डिवाइड रिपीटेडली एंड फॉर्म ट्रिपल endosperm tissue. The cells of this tissue are filled with the reserve food materials and are used in the nutrition of the developing embryo. In the most common type of the endosperm development, the primary endosperm nucleus undergo successive nuclear division to give rise a free nuclei free nuclei. This stage of the endosperm development is called as the free nuclear endosperm. Frequently, subsequently, cell wall formation occur and the endosperm becomes cellular हो जाती है. The number of the free nuclei formed before the cellularization varies greatly. The coconut water from the tender coconut that you familiar with is nothing but free endosperm, free nuclear endosperm made up of the thousands of the nuclei and surrounded by the white kernels is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, that is a pea, groundnut, and bean. Pea, groundnut, bean before seed mature, or it may be persistent in some, such as the castor and the coconut में endosperm persistent हमें देखने को मिलता है. Endosperm is persistent in cereals such as wheat, rice, and maize. Wheat, rice, and maize में endosperm persistent होता है. Completely consumed किसमें हो जाता है? Pea, groundnut, and bean में. Embryo. Let me finish this topic, then I will go for sleep. So it's 17 minutes time. Embryo development at the micropylar end of the embryo sac where the zygote is situated. Most zygote develop only after certain amount of endosperm form. This is the adaptation to provide assured nutrition to the developing embryo. Though the seed differ greatly, the early stages of the embryo development embryos are similar in both monocotyledonous and dicotyledonous. Figure 2.13 depicted as the stage of the embryo in a dicotyledonous embryo. The zygote give rise to the proembryo and the subsequently to the globular heart shape and the mature embryo. Embryo give rise to the proembryo, subsequently globular, पहले प्रोएम्ब्रियो फिर ग्लोबुलर एम्ब्रियो फिर हर्ट शेप एम्ब्रियो. हर्ट शेप तीसरे नंबर पे फिर मैच्योर एम्ब्रियो. पीजीएच पीजीएचएम पीजीएचएम पीजीएचएम. This is the trick process. पीजीएचएम प्रो फिर ग्लोबुलर फिर हर्ट शेप फिर मैच्योर. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. The portion above the embryonal axis level of the dicotyledons is the epicotyle, which is embryonal axis above the level of the cotyledons is the epicotyle, which terminates with the stipules. Which terminate in the plumule or the stem tips. The cylindrical portion below the level of side uh, cotyledon is the hypocotyle that terminate at its lower end in the radicle or the root tips. The root tip is covered with the root cap. Embryo of the monocotyledonous possess only one monocotyledon. In the grasses family, the cotyledon is called as the stichellum, that is situated towards the one side lateral of the embryo axis. That is situated towards the one side lateral of the embryonal axis. At the lower end, the embryonal axis has the radicle and the root cap enclosing and the undifferentiated sheath called the coleoryza. Coleoryza is the undifferentiated sheath, and the portion of the cotyledon axis above the level of the attachment of the scutellum is the epicotyle. Epicotyle has a shoot apex and the few leaf primordia enclosing a hollow follicular structure called the colectile. Hollow follicular structure is the colectile and differentiated is the coleoryza. Leaf primordia enclosing the hollow follicular structure is the shoot apex in the leaf primordia and the hollow follicular structure called the colectile. अब बात करते हैं seeds की. In angiosperm, the seed is the final product of the sexual reproduction. It is often described as the fertilized ovule. Seed are formed inside the fruit. A seed typical consists of the seed coat, cotyledon, and the embryonal axis. The cotyledon of an embryo are simple, structural, generally thick and hollow due to the storage of the food reserve. As in legumes, legumes. Mature seeds may be albuminous and exalbuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development, that is pea, groundnut, and bean. Albuminous seeds retain a part of the endosperm as it is not completely used during embryo development. Wheat, maize, barley, castor. Wheat, maize, barley, castor are the endospermic seed. Wheat, maize, barley, castor. Wheat, maize, barley, castor are endospermic. Wheat, maize, barley, castor are endospermic. Occasionally, the in some seeds such as the black pepper and the wheat, remains of the nucellus are also persistent. This residual persistent nucellus is the perisperm. Integument of the ovule hard and tough protective seed coat. The micropyle remain as a small core of the seed coat. The facilities of this facility is the entry of oxygen and water into the seed during the germination. As the seed mature, its water content is reduced and the seed become relatively 10 to 15% of the moisture by mass. The general metabolic activities of the embryo sac slow down हो जाती हैं. The embryo may enter in a state of inactivity called the dormancy, or in a favorable condition are available adaptive moisture, oxygen, and suitable temperature. They germinate. The ovule mature into the seed. The ovary develop into the fruit. The transfer of the ovule into the seed and ovary into the fruit process is simultaneously होता है. The wall of the ovary develops into the wall of fruits called the develop into wall of fruits. The wall of ovary develop at the wall of the fruits called the perisperm. The fruit may be fleshy or as in guava, orange and mango, or may be dry as in coconut or the mustard seed. Many fruits have evolved mechanism for dispersal of the seed. Recall the classification fruits and their dispersional mechanism that you have already studied in earlier classes. Is there any relationship between the number of the ovules in an ovary and number of the ovule pattern in the flower? In most plants, by the time fruit develop from the ovary, other floral parts degenerate and falls off. Fruits develop from the ovary, other floral parts degenerate and fall off. Few species such as apple, strawberry, cashew, thalamus also contribute to the food formation. Such fruit are called as the fleshy fruits. Most fruits, however, develop only from the ovary and are called as a as called as true fruits. Although in most of the species fruit are the result of fertilization, they are few there are few species in which a fruit develop without fertilization. Such fruits are called as parthenocarpic fruits. A banana is one such example. Parthenocarpic can be used through the application of growth hormones such as fruits and seedlets. Of seed often severe advantage to angiosperm. Firstly, since the reproduction processes such as the pollination or fertilization are independently water, seed formation is more dependable. Also, seen how better adaptive strategies for the dispersal of new habitat and help for the exercise. 2. Colonize in other areas such as they have sufficient food reserve, young seedlings and nour- nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generally generates new genetic combinations leading to the variations. So guys, I am feeling too sleepy. I will end this session right here, right now. And there is the one, two, three, four paragraphs and summary including five things are left over to study. I will study it in the morning by my own. And for this session, this is I will finish it there only. And after that, I will jump on sexual reproduction in the human reproduction. And with that, these two chapters in biology are coming on the Sunday exam, which I have to be prepared. After theory, I will do some practice questions. And for zoology, I will firstly do the question practice, then I will read the text. Okay, that's all for the today's session. Good night, everyone. Have a wonderful